All right, good morning, everyone, or afternoon. Welcome to your second day of remote learning. Today, for both biology and AP environmental science, you are going to see we're covering, awkwardly enough, infectious disease. It was planned on our curriculum for both classes to be covered this week. I just didn't plan on it being so relevant. Infectious disease is classified as a form of biological risk, and we're going to talk about things like prevention and remediation. Prevention are ways that we can prevent things from getting worse. Remediation is how we can clean up the mess. In biology and in environmental science, prevention is often easier and cheaper than remediation. So we'll be exploring stuff, including what's going on in current events from the perspective of both prevention and remediation. For today's class, please don't volunteer any of your personal medical information to me. Please don't tell anyone in the class whether or not you've been vaccinated as well. When we get into biological risk, it's a disease that's considered to be transmissible, which I'll talk about in the next slide. Other things to talk about with this are ways of prevention, like vaccination, and as well as ways diseases can become more dangerous. We'll talk later on this week about antibiotic resistance. Just so you know, the picture here is a scientist standing at the entrance to the cave where almost every African case of Ebola seems to be traced as the source of origin. That means they think somewhere near this entrance to the cave is where they is probably the origin for Ebola. And of all things, they think bats may also be the likely culprit in that. Now, here's some terminology to talk about with disease. One key thing to know is an infectious disease is one that can be passed on to others. So, for example, like if I had a cold and I gave the cold to someone else, that would be an example of an infectious disease. By comparison, there are other diseases like Alzheimer's and diabetes that you don't actually catch from others, but they just develop, but they're not things you could pass on to others. When we're talking about an infectious disease, it's caused by a pathogen, and that's what can pass the disease to others. So we have great examples. Less scary one is athlete's foot would be an example of a fungus, and that's also a pathogen. Another one that we talk about a lot in Western states that affects older people is this disease called valley fever that actually is a mold or a fungus spread in dust. Later on this week, we'll talk about various worms that can cause disease. One of the deadliest groups of animals or organisms would be the protists, because malaria currently remains the deadliest disease on the planet. And it's caused by a simple single-celled organism. It's a type of amoeba. Pretty soon, we're going to be doing a comparison between bacteria and viruses. Bacteria are cells without a nucleus. Viruses are acellular parasites. And if you look over here, these aren't to scale. These are very tiny little things. We didn't even know they existed until the past century. So an example of this would be HIV and AIDS, which I will record a lesson about the history of the AIDS epidemic in the next week as well. The weird thing about viruses is they're not technically alive, but we do kind of classify them as an organism that's a pathogen. Prions are even weirder. Prions are actually basically just a protein that changes the behavior of a cell. If you've ever heard people talk about chronic wasting disease, that would be a prion. One way to think about this list is once you go from worms, these things get steadily smaller going down the list. Now, one important thing to know is we didn't know much about bacteria or viruses until the 1900s. That creepy picture right there is actually how we perceived our understanding of diseases. They were called miasmas, and it was thought kind of the smell of, gross and filth and rotting things. That was what actually spread the diseases. It was invisible vapors and things floating through the air. And to put disease in perspective for all of you, antibiotics were not used until 1942. We didn't really start understanding their resistance, and those are only used for bacteria. And that seems so long ago to all of us, but as I was researching this, it dawned on me that my dad's family, antibiotics weren't available till my dad was 5. Granted, I'm really old, but not as old as my dad. Oh, so many words on this slide. This is the important thing to know between bacteria and viruses. And this ties in with taxonomy or classification of life. Bacteria include things that are single-celled. They lack a nucleus. Examples include E. coli, gonorrhea, leprosy. They reproduce by division, meaning one cell becomes two cells become four. These can be treated by antibiotics. So if you've ever gone on antibiotics for a chest cold, it usually means you've developed some sort of bronchitis that they established was bacterial based. Viruses are acellular and they're not alive. One way to get biologists to start fighting is ask them, is a virus an organism or not? One example that we'll be talking about in the next few slides is COVID-19. I'm also going to be talking about HIV later this week. A virus must infect a host cell, and it'll basically inject its DNA into that, making that host cell do nothing more than make copies of the virus. So some of these can be treated with antivirals. Those, in perspective, didn't exist until I was in high school in the 1990s. So here's the virus life cycle. That little spaceship lander thing is actually a virus attaching to a host cell. It injects its DNA, which is that little blue streak, and it goes into the DNA of the host cell. In this case, this is a bacteria. And here you see it's actually now inside the host cell. This stage is called integration. The weird thing that's going to happen is this little strand of DNA is basically the virus's active party. And what it's going to do is it's going to make that cell start mass producing copies of that virus. Here, this diagram's only showing through 3. Generally, what's going to happen in all of these is most viral infections is a host cell will produce thousands, if not hundreds of thousands of copies. And often that host cell will destroy itself, making copies of that virus till the cell is destroyed. And this is one of the main things to understand with viruses. So while one bacterial cell can become 2, which will become 4, in a virus which is significantly smaller and can only be seen with an electron microscope, one virus can infect a host cell and make thousands, if not hundreds of thousands of copies. So here's the difference. Bacteria reproduce through fission, they double. Viruses are just constantly making and chugging out tons. Now let's talk about COVID-19. It's in a group of viruses called coronaviruses because they are shaped like the sun or a crown. We call it an emergent disease, meaning that it has recently appeared. It's zoonotic because it probably originated in animals near Wuhan, China, most likely at a food market. Most viruses often have, the ones that impact humans, or have origins in other organisms. If you heard people talk about H1N1, it's often called swine flu because it originates in pigs. We have other strains called bird flu or avian flus. In the case of HIV, we've found its source of origin in chimpanzees. So zoonotic means it originates in a different group of animals, zoo meaning animals. You're going to hear newscasters say zoonotic, but technically you're supposed to say zoonotic. So it's an emergent disease meaning new. This, by the way, is a picture of how the virus looks. It is not this big. You could not see it with any microscope we have access to in a high school. Oh, I almost forgot. COVID-19, the 19 means this virus was actually recognized by the World Health Organization in 2019 as its time of origin. So when we talk about viruses, we have a few things to talk about. One thing is the infection rate, which is R naught. R naught of 1 would mean that if one person has it, they're going to probably give it to one other person. R naught of 1.2, this is where the math gets weird, means if 5 people have it, those 5 people will collectively pass it on to 6 more people. The bigger the R naught, the worse things are. So examples of R naughts are Ebola is an R naught of about 2. HIV is an R naught of about 4, meaning someone with HIV is statistically going to probably pass it on to 4 people. This number, so you know, has actually changed, and you'll find out about this. It's significantly lower now. It's below 1 with access to healthcare. SARS, which stands for sudden acute respiratory syndrome, this one has an R naught of 4. It was highly contagious. Mumps, which has been going around Colorado, sucks, and its R naught is 10. Measles, which we will talk about measles this week, has an R naught of 18. So in December, when a few yahoos went through the airport with contagious amounts of measles, if you imagine someone with an R naught of 18 going through an airport, one reason why measles is so contagious is the virus can live for a long, or sorry, I think, is this a bacteria? I think it might be a bacteria. I forgot. It's highly contagious. And so now, here's a lot of adults are saying it's just a really bad flu. The truth is we don't know. There's a link right there, and you can get this through the website. The R naught we think currently of COVID is around 2.2, but we're not sure. R naught of flu is around 1.1 to 1.4. Just looking at that, COVID-19 is actually more contagious. Now, the reason why I'm saying we're not sure is we're now realizing people who don't have symptoms can also pass it on. People, if they don't have symptoms, probably haven't been tested, which means we also don't know how many people could be passing it on, which is why every adult is kind of having panic attacks right now. But this is also why you get to sit at home and learn in your pajamas. Mortality rate for the two, mortality rate is how many people die. For influenza, mortality rate is generally less than 1. With one exception being the 1918 flu. We don't actually know the mortality rate of COVID-19. And I know some of you are getting frustrated with that, but we don't know a lot right now. A vaccine is available for influenza. And you'll often hear people talk about influenza getting vaccines. I get them every year just because I have some very old parents, and if I have the flu and risk bringing it to them at their age, it could be fatal. Tamiflu can actually treat the flu if you have flu-like symptoms and you go and get a flu test and get medical treatment within about 48 hours of your first symptoms. We believe the COVID virus actually originated in bats. Bats seem to be a pool of really bizarre diseases that are crossing over into humans. Bats are colonial. If you've ever seen a bat colony, they are nesting and cuddling together more so than you see more contact than those spring breakers that were in Florida last week. One other issue with bats is when they fly, their body temperature becomes higher than almost any other mammal. Most mammals, including humans, fight off viruses with fevers. In the case of bats, because they have such high body temperatures, a virus that can survive their body is often hard for humans to fight off. Another thing to know is how do we name the viruses? In old days, we named them after the animal of origin, such as swine flu or avian flu. We also used to name them after their place of origin, but we've kind of phased that off because often we were wrong. And I'll give you an example, the next one. In the 1980s, we began changing how we name diseases because of stigma and prejudice. Stigma is kind of shame and judgment, inferring that disease means someone must be filthy, and prejudice meaning we're just going after a group of people. In the early 1980s, HIV and AIDS was referred to as gay-related immunodeficiency or gay cancer. We didn't change the name to human immunodeficiency virus until the first straight people started getting contracting HIV, or at least getting diagnosed. Now, here's an example of when we really bombed a disease naming. The 1918 flu was incredibly fatal. Here are some pictures of people sewing extra masks, which is actually something that people are encouraged to do right now to help those fighting the disease. Initially, it was called the Spanish flu because the first cases were found in Spain. Then it was called the swine flu. One reason why this flu was disastrous is it flew all around the world. What we now understand is during the First World War, this was going through military camps throughout Europe. And then everybody went home and brought it home. It was called the swine flu because the flu strain is the structure of one found in pigs, which means at some point, just like the movie Contagion, somehow like a human got exposed through broken skin or whatever to a pig virus, most likely in processing of meat. So we now refer to it as the H1N1 flu. Awkward bit of history is the Spanish flu or 1918 flu. What we now understand probably originated at a military barracks in the southeastern United States, I believe near Alabama. So if you wanted to be accurate, you could have that. And I'm just letting you know this to know that it is not appropriate to refer to this flu as the Chinese flu. Also, just so you know, the deadliest disease currently is malaria. Malaria affects about 250 to 500 million people a year. It's caused by an amoeba that is spread by mosquitoes. The way we treat malaria are two ways, both focusing on prevention as the ideal thing. If you've ever had tonic water, it's disgusting. The thing with tonic water is it contains an extract named quinine. Quinine is also called the fever tree of all things, and malaria causes a fever. About 130 years ago, they discovered if you had small amounts of quinine in your bloodstream, which you could get by drinking quinine in water, and by the way, water with quinine is called tonic water, tonic meaning medicine, it would reduce your chances of getting malaria. One weird thing is tonic water tastes so bad, people were willing to face death than to have the taste of tonic water until someone discovered if you cut it with a little bit of gin and squeeze some lime in there, it tasted delicious, which also means the mosquito invented the gin and tonic. Just so you know, this will no longer protect you from malaria because through, of all things, natural selection and evolution, the amoeba is now resistant to this amount of quinine. So now you usually have to go to a doctor and get prescribed malaria pills if you're at risk of malaria. So, but cases of malaria have decreased for many years. One reason why is we found treatments like quinine. Another reason is even though DDT has problems in some areas, DDT is one of our best last defenses against mosquitoes that spread disease. Also, draining wetlands. These have environmental problems. But this is examples where these are all examples of using prevention to treat the disease. One challenge is sometimes one solution causes other problems. In the case of malaria, what we're actually seeing is, Florida has decimated their natural ecosystems. These blue areas over here used to all be wetlands. Now they're not. One benefit of this is that they have reduced the risk of malaria in Florida. The trade-off is this is now causing problems with water quality and loss of wildlife habitat. So other update is these days, most doctors would prescribe a stronger medication like Malarone. And what these medications like quinine do is it actually puts a trace amount of these in your bloodstream, and that actually makes your blood too toxic for the amoeba to survive. If you've been watching the news, you're hearing people talk about how some malaria medications, those are the treatment ones, not prevention ones, might be able to help save people who've been infected with COVID-19. Those are what we call anecdotal, where people took the pills and survived. We don't know if it was the pills that caused the survival or if those people were about to get over the disease anyhow or had a mild infection. Currently, the country of Nigeria has actually had in the past few days a bunch of people overdose because they heard that those medications could treat malaria. I'm not saying could treat COVID-19. Currently, those are going through medical trials to see if they are effective. But often you'll hear someone with no science background say, I heard something is a treatment, and it can actually cause greater problems. Here are some examples of other emergent diseases. And of course, we can now add COVID-19. SARS is the one that actually originated in China as well. Zika's place of origin, I believe, is still under discussion. We'll be talking more about HIV in the next lesson. HIV and Ebola both originate right here. MRSA is one we'll talk about with antibiotic resistance. And once again, the process of a disease jumping from one animal into the human population is called zoonosis. And another thing to know is diseases are going around the world faster than ever because humans now fly. Not like with our own power, but with the help of airplanes. In the old days, if you had a disease, say right here, it stayed isolated. But now someone can be wandering a food market here and fly back to Seattle and then get onto another flight and go somewhere else. So this is why diseases are traveling faster than ever. And going with this, how do we deal with outbreaks? So treat the disease if possible is one of the first goals. And when we treat the disease, we deal with things like remediation, which is treating someone who's infected. Vaccination or other forms of prevention are also used because they're cheaper and easier. In this case, for COVID-19, we don't have a vaccine yet, which is why you're hearing so many people talk about social distancing. We also want to find ways to contain the disease. Right now, you are all staring out windows, wondering why you can't hang out with anyone. Right now, we're in the middle of trying to contain the disease because vectors are the things that spread the disease. In the case of COVID-19, humans are vectors, and we're now realizing humans can be vectors who aren't contagious. So to go with treatment of disease, a treated disease is generally less deadly. It is also less contagious. And throughout the world, we have some major challenges with treating disease. Not everybody has access to healthcare. Treatment can often be very expensive. In the case of COVID-19, we're not sure how to treat it yet. And on top of it, if you watch anything on social media, you'll see tons of stigma about it. Vaccines, which we don't currently have for COVID-19, we do have for other things. And the goal for these is herd immunity. If you imagine going back to being in my class, if everybody were vaccinated for measles, no one in the room would ever get measles because they were immune for it. If we only had 90 to 95% vaccination rate, so let's say one person in that room wasn't vaccinated for measles, only one person would be able to get measles in that room. They wouldn't get it from anyone in the room because the rest of us would be vaccinated. So that is called herd immunity. And vaccinations are highly effective at preventing a lot of infectious diseases. One weird thing in Colorado is we have one of the highest rates of lack of vaccination in the country. Important thing to know is the more people that are vaccinated, the less contagious the disease comes. And that's what you see in this model. Here you see the disease spreading rapidly through a population with 0% vaccinated. The red showing, or blue being people who aren't vaccinated. Yellow dots show people who are vaccinated. As we switch from blue people, unvaccinated people, to people who are vaccinated, you're seeing that the disease stops spreading. And once you get to 95% vaccination rate, we've achieved what we call herd immunity. Importantly to understand is not everyone can be for medical reasons, can be vaccinated. Here's our challenge to vaccines. People take medical advice from Jenny McCarthy, whose claim to fame was being married to Jim Carrey and a host on an MTV talk show. She had no science background. The anti-vaccine movement is problematic because it has increased risk for everyone. It's based on research that was actually retracted. The article that everybody quotes was published a while ago in the British Journal of Medicine called The Lancet. Within a year, they retracted it. The basis of the study was saying that vaccines cause autism. In this question, our author argued that when someone goes in for their vaccines around the age of 3, shortly afterwards, a lot of parents commented that right after they got the vaccines, they became autistic or were diagnosed with autism. The reason why that actually happened is it's right around age 3 doctors are actually are looking at their patients, the little 3-year-olds, and around 3 years old is when you start noticing whether or not there's manifestations of autism. Manifestations meaning something is showing up. It's right around the age of 3. They start noticing a little kid is not making the right eye contact or is a little fussier than others. It was not because they got the vaccine. It was because at age 3, that kid was making eye contact with a doctor who was noticing these things. One problem with people going around quoting the science that actually was retracted. Retracted means the journal Lancet apologized and said it was a flawed study and they can't support it. It has led to global outbreaks in the US, or outbreaks in the US as well as globally, of mumps, whooping cough, and measles. Uh, Even weirder, they're now realizing measles, if you get measles, might actually wipe out your vaccine history. Um, in my case, my doctor actually made me redo all my vaccines because our vaccination schedule is based on people not taking medical advice from Jenny McCarthy. But since we have problems. Here's an example of a disease we could wipe out, but we haven't: polio. And you'll hear some people say, well, it's great to not be vaccinated because it builds up your immune system. This is what polio looks like a few years later. A lot of people with polio lose permanent full use of limbs. Another example of this is this megachurch. Their pastor told everybody to not get vaccinated because it was against their religious beliefs. Some of them did relief missionary work in India, where they picked up measles. Then some of them flew to Disneyland, and guess what they brought with them? And the challenge with measles, if you remember that R naught, the R naught for measles is highly contagious. And what people didn't quite grasp is they didn't just go there. People from Disney World or Disneyland then got into airplanes after being exposed to measles and went to all of these other places. So this was a pretty rough outbreak. One of our other goals for a disease is eradication, which is to wipe it out. This is only possible if there's no reservoir of living animals for a disease. So in the case of Ebola, they believe its reservoir is primarily this cave, but it seems to be found in bats, just like COVID. And our dilemma is this: we can't wipe out all bats. They serve an ecological role, and we usually can't intentionally wipe out all of something. We usually do it by accident. The other issue with bats is they can fly hundreds of miles and hang out in different colonies. They're very social organisms, so they're flying around almost as much as humans. Other things we talk about are containment of disease. The old-fashioned approach were leper colonies or quarantine things. One challenge with containment is it creates stigma. An example of stigma right now is if you see anyone cough out on the streets, you panic. Where I grew up, this is an example of a horrible containment approach. This was a quarantine island for anyone that had the flu. And just so you know, the water here is 3 feet deep. Anyone that had flu in this remote area all hung out on this tiny little island. That island right next to it was called Rum Runner Island because that is where a lot of bootleggers were distributing alcohol. So this was an example of really failed containment. When we have a vector that is non-human, we try to control that vector. So in the case of mosquitoes, you'll often see this in the summer. To deal with things like West Nile virus, we will spray for mosquito populations. It has some environmental risks, but they're trying to balance everything but preserve human lives. For dogs and cats, they often encourage you to give them flea or tick drops because your dogs and cats can carry fleas, or in this case, this gross little thing is a well-fed tick. Some ticks can actually jump off of your pets and infect you with, say, Lyme's disease. Another thing is avoiding animals that can spread zoonotic diseases. So most people that don't work with bats intentionally avoid bats, not just because they look creepy. If you look closely, they actually look really cool. But another reason is the more we can avoid those animals, the less risk we have for those transmissions. This is one reason why I actually do wear mosquito repellent when the mosquitoes are bad, even though it might have some other problems. With slaughterhouses, the USDA in the US, the US Department of Agriculture, enforces pretty tight slaughterhouse policies to protect both the workers and the general public. The same applies for feedlots. And throughout the United States, we have very tight policies to reduce the jump of a disease from an animal to a human. Now let's talk a little bit about antibiotic resistance. Actually, I think this is enough for today's class. So I hope you are all well. Remember, social distancing is great. And your assignment is to get off of Facebook and binge watch something mindless on television.